Hi everybody, welcome back to our molecular biology lecture series. We're continuing chapter 5, and in the last lecture I covered some of the basics of molecular biology techniques to study genes and gene function, including molecular separations, labeling molecules with radioactive and non-radioactive tracers, and using labeled nucleic acids as probes in hybridization techniques to detect specific sequences. In this lecture, I'm going to cover topics in DNA sequencing and restriction mapping, and introduce the concepts of PCR-based mutagenesis and protein engineering. In our first topic, DNA sequencing, we will talk about some different methods of DNA sequencing, focusing mostly on classical Sanger sequencing. This is an old technique that has been around for 40 years, but still very widely used today. And we'll briefly touch upon newer techniques of high-throughput sequencing, and I will explain some of the details of Illumina sequencing, which uses similar concepts to Sanger sequencing. As I said, Sanger sequencing is sort of the classical method of DNA sequencing. It was developed by Frederick Sanger, who won the Nobel Prize for his work on DNA sequencing in 1980. This was his second Nobel Prize. His first Nobel Prize was for protein sequencing, and particularly for determining the amino acid sequence of insulin. The main principle by which Sanger sequencing works is through the use of chain-terminating dideoxynucleoside triphosphates. What is that? You will remember that for DNA synthesis, the DNA polymerase uses dNTPs to add nucleotides to the 3' end of the growing strand. So in the DNA molecule, the next base is added to the 3' OH, but if you have a dideoxynucleoside triphosphate, or ddNTP, we're missing the 3' OH. You have no OH on the 3' nor the 2' position of the pentose sugar, so that's what makes it a dideoxynucleoside triphosphate. Once this gets incorporated into a DNA molecule, the next base cannot be added. This results in chain termination. Normally, the next base would be added to the 3' hydroxyl group, but in dideoxynucleotides, this is absent, so no further polymerization can occur. Illustrated here is the classical method that Frederick Sanger developed in the 1970s. Starting with a primer that has been radioisotopically labeled, you can carry out DNA synthesis reactions using DNA polymerase and dideoxynucleoside triphosphates. Where you have chain termination tells you where a dideoxynucleotide was incorporated. You can carry out 4 different reactions that include one of 4 different dideoxynucleoside triphosphates. You will also include the natural dNTPs in this reaction. You'll denature the DNA and then let the primer anneal to the template strand for DNA synthesis. The DNA synthesis is carried out with the natural dNTPs and one of 4 different dideoxynucleoside triphosphates. In this example here, it will be ddATP. If we carry out this polymerase reaction, there is a chance that dideoxyadenosine will get incorporation where the polymerase encounters a T in the template, and that will stop polymerization and terminate the chain. But there is a chance that a normal deoxyadenosine will get incorporated and the reaction will carry on until the next T in the template, and then at that position, there is a chance that a dideoxyadenosine will get incorporated and stop polymerization. Synthesized single strands of DNA, which carry a radioactive label from the primer, are polymerized from many copies of the template, and you have a distribution of DNA fragments of different lengths, where synthesis was terminated by the incorporation of a dideoxynucleotide. You can do reactions like this with each dideoxynucleoside triphosphate and run a high-resolution gel electrophoresis for the reactions, which will separate the fragments by size. This gives you a pattern of radioactively labeled DNA fragments, and by the order of those fragments, you can determine the sequence of the strand of DNA. We see here, for example, that the shortest fragment is one that terminated immediately after incorporation of dideoxyadenosine to the primer. The fragment that is one nucleotide longer terminated with the incorporation of a dideoxycytidine. You can see that the sequence of the synthesized strand from 5' to 3' is A, C, G, C, T, A, G, A, C. To visualize this again, in normal DNA replication, DNA polymerase uses dNTPs to add nucleotides, giving an extended strand that has a 3' OH to which another nucleotide will be added. When you have addition from a dideoxynucleoside triphosphate, a ddNTP, missing its 3' OH, there is nothing to attach the next nucleotide to, so the chain gets terminated. This is illustrated in the figure from your textbook. In the gel electrophoresis of the reactions, you can see different bands corresponding to different bases and positions where termination occurred. The method by which you'd sequence the DNA would be to go through these gels and identify which base is at each position. In the old days, this would be done manually. It would be the work of a graduate student, but nowadays it's all automated. A lot of my senior colleagues who studied in the 1980s would tell me that they'd spend hours interpreting these gels to determine a gene sequence. You can imagine how long it would take to read the sequence of hundreds of bases. But that's the principle behind Sanger sequencing, and now the process has been much more automated and streamlined. Now, instead of using radioactively labeled primers, we have fluorescently labeled bases that we add onto the synthesized strand. This is called the dye terminator method. Instead of a radioactive label on the primer, we have fluorescent labels on the dideoxynucleoside triphosphates, so that the synthesized fragment incorporates the fluorescent label on the 3' end. Generally, these dye terminator ddNTPs look like this. They have attached to the base a fluorescent dye. Each base, A, T, G, or C, has a different fluorescent dye, shown here. These emit at different wavelengths, allowing you to distinguish which base was incorporated so that you can do the chain-terminating DNA synthesis reactions with all 4 ddNTPs together instead of having separate reactions. The fragments that are fluorescently labeled can then be separated according to size by capillary gel electrophoresis, and the fluorescence detected by laser excitation, producing a chromatogram that can distinguish each of the bases according to their label. The sequencing is typically done in an automated process. The chromatogram has different peaks corresponding to different wavelengths that can be used to identify the bases, and a computer can automatically interpret this to determine a DNA sequence. You can usually sequence about 1,000 bases starting from a primer before the resolution of the chromatogram drops, and you can no longer distinguish bases at specific positions. You can perform this in parallel and do Sanger sequencing for large numbers of cloned DNA molecules. You can do dozens of these sequencing reactions with dye-terminating dideoxynucleoside triphosphates and run the electrophoresis in parallel on the same sequencing instrument. So you can sequence lots of DNA clones simultaneously. But this is not quite the same as the high-throughput sequencing that we're going to talk about. There are a couple of methods of high-throughput sequencing. Your textbook talks about pyrosequencing and Illumina sequencing. But in the last decade or so, a number of newer alternative methods have been developed for high-throughput sequencing. Some of these are listed here: solid sequencing, ion torrent sequencing, Biol sequencing, and Oxford nanopore sequencing. However, we'll focus our discussion on Illumina sequencing, which is very widely used and follows similar principles to what we discussed with dye-terminating Sanger sequencing. One major distinction is that in Illumina sequencing, the chain termination from the 3' modified nucleotide can be reversible, and that the fluorescent tag can be removed. The sequencing is still based on a templated polymerase reaction extending synthesized DNA, then measuring fluorescence on the modified bases that are incorporated. But the difference is that these modified bases terminate DNA synthesis by a mechanism that is reversible. Rather than the OH group being removed completely from the chain terminating nucleotide, it is instead blocked by a removable protecting group. And you also have a fluorophore here, which can also be removed. After the incorporation of one of these modified bases, you can measure the fluorescence and tell what the identity of that base is, because you have different fluorophores for each base, and then after you measure the fluorescence, you can chemically cleave off the protecting group and the fluorophore, and then you're ready for the addition of the next modified base. How does this process work in high throughput? You might have genomic DNA or some other very long DNA that you want to sequence. You can fragment it into smaller pieces. This fragmentation can be done by physically breaking up the DNA by mechanical force, or it could be done chemically or enzymatically, using partial DNase digestion, for example. You get a bunch of different small fragments, usually that are less than 500 base pairs, and then you can ligate these to adapter sequences. You end up with short DNA fragments ligated to adapter sequences. You want to have these fragments with two different adapter sequences at the ends. Sometimes you will get fragments that have two of the same adapter sequence on both ends, but you can enrich for the fragments that you want with the different adapters at each end. I won't get into those details here, however. You can then anneal these fragments by their adapters to oligonucleotide primers that are immobilized to a surface. The primers are complementary to the sequence of either adapter, and the DNA fragments to be sequenced will anneal at different spots on the surface. In the next step, you can perform PCR with these immobilized primers, and because they are fixed in place, what you end up with is these fragments of DNA are copied in clusters on this surface. Each cluster has identical PCR fragments that are separated spatially from one another in clusters on this surface. So each cluster corresponds to a different sequence with many identical copies of a DNA fragment within a given cluster. When you perform Illumina sequencing, the DNA in each cluster serves as a template with a primer annealing to one of the adapters. DNA polymerase adds the modified bases, the reversible di-terminator nucleotides. After addition of one of these, you can measure the fluorescence of the modified base incorporated into synthesized DNA annealed to its template at each cluster, which corresponds to a fragment of the genomic DNA. You can take an image and tell what color fluorescence is observed in spots which correspond to specific bases incorporated into different clusters of identical DNA fragments. After you have done the measurement, you can chemically cleave off the blocking group from the 3' end of the synthesized DNA and cleave off the dye. Then you can carry on with the next base addition and measure again the fluorescence at the spots corresponding to different DNA fragments. We can continue this process, and at many different spots you can sequence these short fragments of DNA. When you have all that information, you can piece together the total sequence from a genome, for example, from the sequence of many overlapping fragments. These correspond to different clusters on this surface, which were sequenced through a series of images of fluorescent spots through multiple cycles of addition of modified bases. Usually, before you do sequencing, there are some other methods that you can perform to get a crude analysis of your DNA. You can do some preliminary mapping. Here we will talk about restriction mapping. You can do restriction digestion and locate restriction sites as landmarks on your sequence of DNA. This could give you some quick information of the organization of the DNA sequence before you send it off for sequencing using Sanger sequencing or high-throughput sequencing. For example, say you cloned a piece of DNA into a vector. You might want to do restriction mapping to quickly check if you have the right DNA in your clone. You can use this method to confirm that the restriction map matches what you should get from the correct sequence. In this example from your textbook, you have a fragment of DNA cut with HindD3 that you want to clone into your vector that has been cut with the same enzyme. You also know that your vector has another restriction site here called EcoR1. You know that the distance between HindD3 and EcoR1 sites on the vector is 0.3 kb. In this example, the DNA fragment that you want to clone has another restriction site here, BAMH1, which is closer to one end, 0.4 kb, than the other, 1.2 kb. So you can insert this fragment into the vector, but it could be inserted in two different orientations. Say you want it to be cloned in one orientation and not the other. You can check the cloning orientation if you isolate the DNA and digest it with restriction enzymes and then analyze by electrophoresis the fragments that are generated from the digestion. In this example illustrated here, you are cutting with BAMH1, which has a recognition site inside the insert, and EcoR1, which has a recognition site on the vector. In one scenario, the DNA was inserted in this orientation, and digestion with BAMH1 and EcoR1 gives these fragments that are 3.6 and 0.7 kb. From the size of the fragments, you can work out that for this clone, your insert has been ligated so that the BAMH1 site is closer to the EcoR1 site, compared to the alternative scenario, in which the fragment was inserted in the opposite direction, where your BAMH1 site is further away from the EcoR1 site. In that case, BAMH1 and EcoR1 digestion would give fragments that are 2.8 and 1.5 kb. In another example here, say you want to get a map of the organization of this 17 kb plasmid. You can digest it with a couple of restriction enzymes. For example, when you cut it with HindD3, you see that you get two fragments, 6 and 11 kb. And when the 17 kb plasmid is cut with PST1, you get two fragments, 5 and 12 kb. For a circular plasmid, you know it must have two HindD3 recognition sites and two PST1 recognition sites. If you cut the plasmid with both HindD3 and PST1 enzymes, you will get four fragments, 7 kb, 4 kb, 5 kb, and 1 kb. So you want to piece together a map of these restriction site landmarks. You know that for HindD3, these two sites have to be 6 kb apart. You also know that for PST1, those sites have to be 5 kb apart. So can you reconcile the information from the HindD3 digestion and the PST1 digestion with this information here showing that digestion with both enzymes gives you these four fragments? What you can do is draw a map showing the positions of HindD3 sites relative to one another, and another map showing the positions of PST1 sites relative to one another. Then you can try to overlay these maps to get a complete map. You can use some of this information. For example, we know that we get a 1 kb fragment from the double digest that is not in any of the single digests. So we know that we have to have a PST1 site that is 1 kb away from a HindD3 site to give us a fragment of that size. We also know that compared to the single digest with HindD3, when we digest with HindD3 and PST1, both of these HindD3 fragments are split into smaller fragments, since we don't see either a 6 kb fragment nor an 11 kb fragment. So we could try putting the PST1 site 1 kb away from the HindD3 site in this direction, and place the other PST1 site 5 kb away on the other side of the HindD3 site. But this would not result in fragments of the correct size. So we can try the alternative. With a PST1 site 1 kb away from this HindD3 site on the other side, and the second PST1 site 5 kb away from the first on the opposite side of the Hindi 3 site. This would give us fragments matching what we see from gel electrophoresis of the digestion. You can also improve this process by using southern blotting to find out which fragments overlap with one another. In this example here, you have a linear piece of DNA that you can cut with either EcoR1 or BAM-H1, giving you different fragments that you can separate by electrophoresis. How can you tell which of these fragments are overlapping? You can isolate some of the fragments from one digest, label them, and then use them as probes for southern blotting on a gel from the other digest. Here in this example, this gel from an EcoR1 digest is blotted using a labeled 12 kb fragment from a BAM-H1 digest as a hybridization probe, showing that it overlaps with the 12 kb and 6 kb fragments from the EcoR1 digest. Similarly, this 8 kb fragment from the BAM-H1 digest can be labeled and used as a hybridization probe, and from the southern blot we can see that it overlaps with the 12 kb and 3 kb fragments from the EcoR1 digest. So far in this lecture, we have been talking about analyzing DNA sequences through mapping and sequencing methods. In this part, I will talk about methods for making changes to the DNA sequence. This is site-directed mutagenesis. If our aim is to study protein coding genes, you may want to make a change in the sequence of a protein to study the effects of some amino acid substitution on that protein. You can make changes on the DNA level of a gene encoding that protein. From a mutant version of the gene you've cloned, you can express a mutant protein and then study the effect of those changes in the protein sequence. So, in general, this is called site-directed mutagenesis. This technique was developed by Michael Smith, pictured here, who was awarded the Nobel Prize in Chemistry for this work. He received this prize in 1993 along with Kary Mullis, who developed PCR a couple of years earlier. Through this method, you can make precise changes to the amino acid sequences of proteins that are expressed from cloned genes by changing specific bases on the gene. Shown here is one of a few different ways of doing this. This is a very common method of doing site-directed mutagenesis, which uses PCR-based methods on a gene that you may have cloned into a plasmid vector. That's illustrated in your textbook and shown on the next slide also. So essentially, you have a plasmid vector carrying the gene coding for your protein of interest, and you want to make some nucleotide change on that gene. You would do that using a pair of primers that are complementary to a specific region of the gene, except for the single or few bases that you want to substitute. Using the plasmid carrying the gene that you want to mutate as a template, you can first denature the DNA and hybridize your primers carrying the base substitution. You amplify the mutant plasmid by PCR and then transform your E. coli. Then you can isolate the clone that carries your mutant plasmid. If this illustration here is not clear for you, let me redraw it for you to illustrate the key steps of this process. So you have some cloned DNA, and say you cloned it into E. coli. Your gene that you want to mutate is on the plasmid, which is in E. coli. And one thing to remember is that E. coli methylates the DNA that it replicates. The gene that you want to mutate is on that plasmid, which is methylated. So you isolate that double-stranded plasmid, and let's say this is the gene that you want to mutate. If you have mutagenic primers that, except for the bases that you want to substitute, are complementary to a sequence, some region where you want to introduce your mutation, you can carry out PCR to copy the entire sequence of the plasmid except for the bases you substituted on the primer. In the example from your textbook, the codon for a tyrosine, TAC, is substituted with a codon for phenylalanine, TTC, that is encoded on the primers. So you have these two primers that are complementary to this region of the gene, with the exception of the mutation in the middle. These will anneal to your plasmid where you want to make that mutation, and when you PCR amplify that, you will get a mutant plasmid plus your wild-type plasmid. You've amplified this mutant plasmid. Now you want to isolate the mutant plasmid, and you want to get rid of the wild-type plasmid encoding the wild-type gene. How can we do this? If you originally isolated the wild-type plasmid carrying the wild-type gene from E. coli, it is possible to select for the PCR-amplified mutant plasmid and get rid of the original. As we had discussed, E. coli modifies its own DNA by methylation to protect it from its own restriction enzymes. So the original plasmid that was isolated from E. coli has GATC sites that are methylated so that E. coli restriction enzymes will not cut them. However, there are certain restriction enzymes from other bacteria that specifically recognize methylated GATC sequences and digest those. DPN1 is one such enzyme that recognizes methylated GATC and leaves unmethylated GATC alone. So you can specifically degrade this wild-type plasmid, and the restriction enzyme doesn't touch the mutant. Now you can transform E. coli, and by going through this process, you select for introduction of the mutant plasmid that was synthesized by PCR amplification and is unmethylated. You can make a mutant plasmid encoding your mutant protein in that manner. And here you see an example where we can change one single base to change the codon from tyrosine to phenylalanine. I could change this to any codon that I want. I could change multiple bases in here rather than a single base, as long as I have a complementary region flanking either side, so I can change the phenylalanine to any other amino acid that I want on the mutant protein. To recap what we've discussed, if we clone a gene, we can make a nucleotide base substitution by site-directed mutagenesis. And through introducing that substitution, we can change the amino acid sequence of the protein that we would express from an expression plasmid. I just showed you a common PCR-based method for doing this, using a pair of complementary mutagenic primers. After getting rid of the wild-type DNA by digestion with DPN1 enzyme, we can isolate the mutant and use that to transform E. coli and then pick colonies. Once we confirm which of the colonies contain the correct clone coding our mutation, something that we do by DNA sequencing, we can then introduce the plasmid into an expression host and produce the mutant version of our protein. That's it for this lecture. In the next lecture, we'll continue to cover methods for studying genes and gene expression.